Cell theory. 1. All living organisms are composed of one or more cells. 2. The cell is the basic unit of life. 3. All cells come from the division of pre-existing cells. Cell membrane and the fluid mosaic model. Function, transport of materials, cell recognition, maintaining cell integrity structure, and homeostasis internal balance. Selective permeability. The membrane is semi-permeable or selectively, differentially permeable, meaning it allows some substances to cross but not others, even if they are the same size. Structure fluid mosaic model, composed of a phospholipid bilayer. Phospholipids have a hydrophilic water-loving head and a hydrophobic water-fearing tail. The bilayer arrangement places the hydrophilic heads facing outward towards the watery environments inside and outside the cell, and the hydrophobic tails facing inward, away from water. Proteins are embedded within the bilayer, creating a mosaic pattern. These proteins have various functions, including acting as receptor sites. The lipid component provides fluidity and flexibility. The embedded proteins provide the mosaic aspect and contribute to the membrane's functionality. Receptor sites. Some surface proteins act as receptor sites, allowing specific molecules to bind to the cell surface via a lock and key mechanism. Protein channels allow lipid-insoluble materials like water to cross the membrane. Channels with ATP transport certain substances, ions, amino acids, glucose across the membrane using energy from ATP, carbohydrates. Chains of carbohydrates attached to proteins, glycoproteins, or lipids, glycolipids, act as identification markers for cell recognition, for example, by the immune system. Nucleus structure. A centrally located organelle surrounded by a double membrane called the nuclear envelope. The nuclear envelope has pores, nuclear pores, and is continuous with the endoplasmic reticulum membrane. Function controls cellular functions, metabolism, growth, differentiation, structure, reproduction. Contains DNA and is the site of RNA production. Chromosomes, structures within the nucleus containing DNA tightly wound around proteins called histones. Involve in DNA packaging during cell division and gene expression control. Nucleolus, dark staining region S within the nucleus. Site of ribosomal RNA or RNA synthesis, a key component of ribosomes. Endoplasmic reticulum, a structure, a system of membranous tubular canals extending from the nuclear envelope throughout the cytoplasm. Rougher, studded with ribosomes, site of protein synthesis. Smooth endoplasmic reticulum sphere, lacks ribosomes. Synthesizes lipids for membranes and hormones, detoxifies drugs and chemicals often involving peroxisomes. Also synthesizes phospholipids and cholesterol, key membrane components. Ribosomes, composition, RNA and proteins. Structure, to non-identical subunits. Assembly, rRNA produced in the nucleolus, combined with proteins, then migrates to the cytoplasm for final assembly. Function, protein synthesis often attached to the RER. Polysomes, polyribosomes, location, free-floating in the cytoplasm. Function, synthesize proteins for use inside the cell. Structure, clusters of ribosomes producing the same protein. Golgi apparatus, structure, stacks of flattened, membrane-bound sacs, cisternae often connected to the membrane. Golgi apparatus function, modifies, assembles, packages, and secretes substances. Receives newly synthesized proteins from the sorts and labels them, and then transports them to their destinations within the cell or to the cell membrane for export exocytosis. The Golgi is composed of stacks of flattened sacs, cisternae, each containing enzymes that modify proteins as they pass through. Vacuoles and vesicles. Definition: membrane-bound storage compartments. Vesicles are small vacuoles. Formation: 1. Pinching off from the Golgi apparatus. 2. Endocytosis of the cell membrane. 3. Extension of the membrane. Composition: phospholipid bilayers. Function, transport and storage of materials. Lysosomes, origin, specialized vesicles formed by the Golgi apparatus. Functions 1. Cellular digestion, for example, of bacteria and viruses. 2. Autophagy, breakdown of damaged cell components. 3. Autolysis, breakdown of the entire cell, suicide sacs. Mitochondria, size, largest organelles and animal cells after the nucleus, shape, sausage-shaped or filamentous. Structure, double-layered membrane, inner membrane folded into crista where cellular respiration occurs. Function, cellular respiration, converts glucose into ATP, the cell's primary energy molecule. Transport across the membrane. 1. Passive transport, no energy required, follows concentration gradient. A. Diffusion, lipids, CO. O. B. Osmosis, HAOC, facilitated diffusion, other molecules. 2. Active transport, energy required. Often against concentration gradient. A. Active transport, B. Endocytosis. C. Exocytosis, diffusion, definition, movement of molecules from high concentration to low concentration. Energy, requires no energy input though heat can speed it up. Rate influencers, concentration gradient, molecule size, shape, temperature, slower in liquids than gases. Increasing the rate of diffusion. 1. Increase temperature optimally around 37 degrees Celsius. 2. Increase concentration gradient. 3. Decrease the size of the diffusing molecules. Membrane permeability and diffusion, lipid-soluble molecules, steroids, alcohols can diffuse directly across the lipid bilayer. Osmosis, definition, the net movement of water molecules across a selectively permeable membrane from a region of high water concentration to a region of low water concentration through protein channels. Water is not lipid-soluble, solute, particles dissolved in water. Solvent, liquid that dissolves the solute water in osmosis. Osmotic pressure, pressure due to the flow of water from high concentration to low concentration. Increases with a larger concentration difference. Water movement in cells. Water moves readily across membranes through protein-lined pores, while many other molecules cannot. This allows water to follow the law of diffusion. Isotonic solutions, net water movement, none. Solute concentration, same number of solute molecules per unit volume on both sides of the membrane. Effect on cells, cells neither gain nor lose water. Hypertonic solutions, hyper means more than. When a cell is placed in a hypertonic solution, water leaves the cell, causing it to shrivel. This is called crenation in animal cells and plasmolysis in plant cells. Hypotonic solutions, hypo means less than. These solutions have a lower concentration of solute than the cell contents. When a cell is placed in a hypotonic solution, water enters the cell, causing it to swell and possibly burst. Bursting is called lysis in animal cells and creates turgor pressure in plant cells. Facilitated transport uses protein channels in the cell membrane to control the passage of molecules. Highly specific, each carrier protein transports only one type of molecule. Molecules move along the concentration gradient high to low. Requires no energy, similar to diffusion in this respect. Explains how lipid and soluble molecules like glucose and amino acids cross the cell membrane.